Welcome back to the Deep Dive, where we take a closer look at the tiny but mighty players in the world of microbes. Today, we're going deep on Alloscardovia omnicolens.
Sounds good.
And to help us do that, we've tapped into some pretty cool resources. Notebook LM, Gemini, Microbe Wiki, and Microbe Canvas.
Excellent.
All right, so let's get granular. Now, this bacterium, it's one we often come across, but maybe don't give a whole lot of thought to.
True.
So let's start there. Can you give us a quick overview? What is this Alloscardovia omnicolens?
Well, it's a gram-positive bacterium, and it belongs to the Bifidobacteriaceae family.
Bifidobacteriaceae. Wait, isn't that like the good bacteria family, the yogurt superstars?
It is, and that's what makes Alloscardovia omnicolens so fascinating.
Okay, I'm intrigued. So we've got this bacterium with a good family pedigree, but is it living up to the reputation?
Well, that's where things get a bit more complex. It's usually hanging out in the gastrointestinal tract and the oral cavity of both humans and animals.
So far, so good.
But it doesn't always stay put. We've also found it in the blood, saliva, the lungs, even in aortic valve accesses.
Okay, now hold on. Aortic valve accesses, that doesn't sound very good bacteria to me.
Right. It definitely complicates the picture.
It does.
It's often labeled non-pathogenic, but the clinical significance is, well, it's debatable.
Debatable.
Meaning sometimes it seems harmless.
Okay.
And other times it's implicated in infections.
So this little bacterium is keeping us on our toes.
Exactly.
So for our listeners out there, the medical microbiologists, what should they be looking for? If Alloscardovia omnicolens is causing trouble, how does it usually show up?
Good question. Well, urinary tract infections are a big one.
Okay, so that's definitely something to keep an eye out for.
Absolutely. We also see an endocarditis.
Endocarditis, right?
And those aortic valve accesses we talked about.
Right, so those seem to be the main trouble spot.
We do seem to be.
Now let's get down to the nitty-gritty. How do we identify Alloscardovia omnicolens in the lab?
Okay, so first up, the gram stain.
Always a glossy.
It'll show up as gram-positive. Short, irregularly shaped rods.
Short, irregularly shaped rods, got it.
And sometimes you'll see what we call rudimentary branching.
Rudimentary branching. What does that look like exactly?
Think of it as these little incomplete branches coming off the main cell body.
Interesting. So not your typical smooth rod then?
Nope, not quite.
Okay, so we've got the gram stain. How about culturing this bacterium?
All right, so for Alloscardovia omnicolens, it prefers aerobic conditions.
Aerobic, meaning it needs oxygen, right?
Exactly, and it likes a bit of carbon dioxide too.
CO2, got it.
If you're using blood agar, you'll see alpha hemolytic colonies after about 48 hours.
Alpha hemolytic.
Meaning it partially breaks down the red blood cells around the colonies.
Right, right. And does it go on anything else?
Yep, it'll also go on Bucella blood agar.
Okay, good to know. So we've covered where it hangs out, what kind of trouble it can cause, and how to find it in the lab. Anything else crucial about Alloscardovia omnicolens before we move on?
Oh, a couple of things. It's a catalase positive and oxidase negative.
Noted.
And for our antibiotic-minded listeners, it's generally susceptible to vancomycin and resistant to colistin.
Vancomycin susceptible, colistin resistant. Sounds like that fits with the Bifidobacteriaceae family pattern.
Yeah, it does.
Okay, I think we've covered a lot of ground here. How about we test our listeners' knowledge with a few quick questions?
Sounds like a plan.
All right, question number 1. What is the gram stain morphology of Alloscardovia omnicolens?
It's those gram-positive, short, irregularly shaped rods, sometimes with rudimentary branching.
Okay, good. Question number 2. What are the key culture characteristics of Alloscardovia omnicolens?
Well, it prefers to grow in the presence of oxygen. It likes a little CO2.
Right.
You'll see those alpha hemolytic colonies on blood agar after 2 days. And it also grows on Brucella blood agar.
Perfect. Okay, question 3. Where is Alloscardovia omnicolens most commonly found in the human body?
It usually hangs out in the gastrointestinal tract and oral cavity. But remember, it can also be found in other spots like blood, saliva, lungs, and even those aortic valve abscesses.
Right, those abscesses keep coming up.
They do.
Question 4. True or false, Alloscardovia omnicolens is always harmless?
Definitely false. While it's often considered non-pathogenic, remember we're seeing more and more evidence suggesting it can be involved in infections, especially those urinary tract infections.
Tricky little bugger, isn't it?
It can be.
Okay, last question. What are some of the challenges a medical microbiologist might face when dealing with Alloscardovia omnicolens?
The big ones are accurate identification and figuring out its clinical significance, because it can look a lot like other bacteria under the microscope.
Yeah.
And because its role in infections isn't always clear-cut, it can be a real challenge to know how to approach it.
Well, I think our listeners are well equipped now to tackle this microbial mystery. Thanks for joining us on this deep dive into Alloscardovia omnicolens.
And to our listeners, keep those microscopes focused.
There's always more to discover in the world of microbes. Until next time, stay curious, and thanks for listening.